The last topic I'd like to go over is what can go wrong with DNA. And those are what we call the DNA mutations. So write that as your heading. DNA mutations. So what we have up here is the normal DNA, which you probably want to write out for yourself, or the normal RNA, sorry, mRNA. The DNA, remember, gets copied into RNA, so if we have a change in the DNA, it's going to change the RNA. So all of these examples are actually going to show how the RNA is affected. But again, remember, the original mutation happened to the DNA, and we're just showing it in the RNA. So here's the original RNA, AUG, AAG, UUU, GGC, UAA. Codes for the amino acids met, lyse, phi, gly, stop. Right, so that's what it normally looks like. What we're going to take a look at is a specific type of mutation called a substitution. So please write that as your heading. So in a substitution, we're going to have the change of one letter of the DNA to another. So just like when you substitute in a basketball game or a football game, you're trading one player for another, kind of have the same thing going on here. In this case, we're substituting a U for this C in the original RNA. And when you do that, a few different things could happen. So in this first example, even though we changed the C to a U, GGC means glycine, but GGU also means glycine. So there's no effect on that amino acid sequence, which means the protein is not affected either. However, sometimes there is an effect on the amino acid sequence. And there are a couple ways that that could happen. In this one, it got mutated. There we go. Into an A instead of a G. And GG, what was the original one? GGC was that original codon, is now AGC. And that does not code for glycine anymore. Now that codes for serine. That type of substitution in which we change one amino acid is called a missense. So in a missense substitution, you change one amino acid to another. But we could also have a change that creates a stop codon. So in this codon, we substituted a U instead of an A. So instead of reading AAG, it now reads UAG, which is a stop codon. And so the only thing that ends up being coded for is methionine. So this is called a nonsense substitution. And in a nonsense substitution, we change to a stop codon. So these are all three substitutions, 1, 2, 3, but in each case there's a different result. Either no change, switching the amino acids, or in creating a stop codon where it shouldn't be. A few other possible types of mutations. Again, we're still working with this original type of DNA. This time, we're going to take the DNA and do an insertion, which is adding a base, or a deletion, which is taking out a base. So in this first one, they've added in an extra U, which in the DNA would have been an extra A. And when that happens, it created a stop where it shouldn't have been. Okay, so it created a nonsense mutation by adding in an extra base. In the second one, it removed a U which in other words, from the original DNA, it removed an A, and it changed all the amino acids after that mutation. This result in changing more than one amino acid is called a frame shift. And the reason it's called a frame shift is because what is determined as being the reading frame for these codons has changed. So let's just take a quick look at what I mean by that. So the original reading frame read AUG, AAG, get my box around it, UUU, GGC, UAA. Okay, those are my reading frames. You can think of them like picture frames. But down below, when we added or took away the U, AUG was still there, AAG was still there. We removed a U, so instead of having UUU, now the next codon is UUG, so that frame changed, which caused every frame down the line to also change. So no longer GGC, now we've got GCU, and then AA is going to go with whatever base is next. So it's going to shift everything after that particular mutation. So we call that a frame shift mutation. The last one down here at the bottom, let's do an erasing here. Takes out more than one base. And this does not cause a frame shift, but it could cause an extra or missing amino acid depending on if 3 bases are added or if 3 bases are subtracted. You can see in this one, it took out a whole set of bases. So we still have methionine, and we still have the phenylalanine and the glycine, but we're missing the amino acid that came between them. So this one here is missing. If we look back at the original one, the one that's missing is this is the lysine. So the lysine was supposed to be there, coded for by AAG, but now it's missing. So again, deletions or insertions, adding or subtracting bases could have a lot of different effects on the mRNA itself. It could cause a nonsense again, so a stop codon. Could cause a frame shift with a missense, so lots and lots of amino acids change. Or it could just take out an entire amino acid, or add in an entire amino acid if it's 3 bases at a time. All right, the last thing I'd like you to do for this set of videos is to write down these chapter quiz questions that I'd like you to look over. Because we're not going to be discussing this information about DNA hardly at all in class, because it's mostly review, I do want you to check yourself with these quizzes. It's chapter 16 and 17, these particular numbers. You know where the website is to log in. What I'd like you to do is actually print your results, so when you're all done with your quiz and you've checked to see which ones you have correct and which ones are incorrect, print off those pages and then hand those in when those are done, so that I can see which questions students are still struggling on. I will give you a due date for printing these out in class.